And then you have to have a linker technology where you have to take that active portion of the cytotoxic drug and have a linker which is stable in circulation, but once it gets into the cell, it should be able to get cleaved. And so there are a couple of ways you can do that. So you can have a linker which is acid labile, so that is, if the pH goes down to around pH 5 or 5.5, so that the linker gets cleaved, and so the cytotoxic drug is released. So when the antibody-drug conjugates are going through the endosomal pathway, so usually the endosomal pH, the early endosomal pH is slightly acidic, and so you could use that environment to design your drug so that the linker that you are designing gets cleaved at the lower pH, and so the cytotoxic drug is released. But it was found out that from a lot of experiment, the acid labile linkers are fairly unstable in circulation, and so now people are moving more towards peptide linkers. So these are so usually it is a citrulline valine kind of linker, so which gets specifically cleaved by an enzyme called cathepsin B. So cathepsin B is present in the lysosomal compartment of the cells, and so when the antibody-drug conjugates get internalized and when they go through this endosomal and lysosomal compartment, in the lysosomal compartment, so this if you have a citrulline valine linker attached to the cytotoxic drug, so then the cit-val linker get cleaved by the cathepsin B in the lysosomal compartment, and the released cytotoxic drug comes out of the lysosome. Either it can go to the nuclei and bind to the DNA, or if the cytotoxic drug is a tubulin inhibitor, it will go in the cytosolic compartment and bind to the tubulin and inhibit the thing. So that's where it can the thing. So the design of the linker is very critical. So the one critical parameter one has to consider here is, so the linker has to be stable in circulation, but should be able to get specifically cleaved inside the cell, either due to the lower pH or due to some of the proteases that are present in the lysosomal compartment.
Thank you very much for that answer.